Richmond.
Yan si Al.
Hi, Alor.
Hello, sir.
Kumusta na?
Ito.
Hi, Al.
Lockdown na naman kami, sir.
Long time no see, Al.
Kumusta na?
Yes.
Naman naka-organize kayo ng presentation. Kahit hindi kayo nagkikita.
Nice to set.
Oh, napagalitan kami.
Sabon ba? Sabon?
Oh, hindi man lang binandawan. Pero tanda-tanda ko na napagalitan.
Double down on our efforts to catch up with science and technology and innovation. Science and technology and innovation, these are indispensable in the modern economy, in the modern world.
The problem being that, while we have our PhDs here, there aren't enough of us. And everywhere else in the world, for research to be successful, you need a complete research group.
It's very important that the PAASE members and scientists based in the Philippines are continuously in contact, so we will know the needs of the Filipinos and opportunities that the Filipinos can have to do science with their PAASE counterparts.
I think that is one of the good programs that they keep on supporting some of the initiatives of our researchers and scientists here in the country.
The role of this cooperation among these government agencies really is to be able to attract science and technology global-based companies to set up shop here. And that is really key in terms of developing the science and technology innovation ecosystem in the country.
We really need to ratchet up the appreciation, the recognition of the importance of science and technology and innovation.
And we are all trying to attain what we call the common good, the greater, higher, long-term good of the Filipino people.
Thank you. Welcome, everyone. Good morning. Okay. Good morning, everyone. Welcome to the PAASE webinars, REC7. It's like 8 o'clock in the morning here in the Philippines. Magandang umaga, magandang tanghali, magandang hapon, magandang gabi sa lahat ng participants from all over. I'm Elmer Estacio po from the National Institute of Physics, UP Diliman, and together with...
Morning, guys. I'm Percival Almoro from UP Diliman as well. Maganda to, Tux, dahil yung PAASE webinar is across disciplines, di ba? So, engineering, physics, biology, etc.
Okay, so right now we have 3 speakers for today's webinar. We have someone coming from the National Institute of Physics to talk about optics and how to use light as a way of using as a special way of light as a noble way to image and interpret things. And we have a former colleague of ours from the National Institute of Physics, who's now in the US, Cornell University, doing work on biophysics. And we have from the younger generation, dati ho naming estudyante, and she's now doing postdoc work at Riken in Japan. And we'll be talking about a novel technique on atomic level imaging. Okay, so I think we're about ready to start, if it's all right.
All right.
Hello, Suzette. Okay, so first off, we have our speaker from the National Institute of Physics, who will also be co-hosting today's webinar. Dr. Percival F. Almoro is a professor of the National Institute of Physics, College of Science, University of the Philippines, Diliman. Salamat ba sa pagpapa-unlock? Dr. Almoro is my colleague at the National Institute of Physics, and at the same time is also with the Office of International Linkages, Diliman. He had his postdoc fellowship at the University of Utsunomiya, Japan, in the most recent one in 2014. Earlier back at Riso National University in Denmark from 2008 to 2009, and much earlier at the University of Stuttgart in Germany in 2006. He's a senior member of the Optical Society of America and is currently serving as an editor for the Applied Optics Journal. Dr. Almoro's main research interests are in holography, phase retrieval, and interferometry. So without any further ado, let's begin into the optics world of Dr. Percival Almoro. Anytime you're ready, Val.
Thank you, Professor Estacio. Good morning and good day to everyone. So I'll be sharing my work on randomized light for enhanced wavefront reconstruction, principles and applications. So this will be the flow of my presentation. First, I'll give an overview of the basic principles to understand this technique, namely wavefront reconstruction and phase retrieval. I will then focus on the technique that we've been working on perhaps in the last 15 years, which is called the multiple plane and diffuse illumination phase retrieval. And then I'll discuss again an important, just to emphasize this important property of light, which is the randomized and diffuse light, which paves the way for this technique that we've been working on. And then I'll touch on the two approaches that we've been doing, experimental and algorithmic strategy or approach. Particularly in experimental approach, we use a diffuser projected on an active optical device, which is the SLM. And for the algorithmic or computational approach, we use a randomized sequence for the reconstruction of light. And then I'll mention directions for future work. All right, so wavefront reconstruction is a two-step process. So first you record the information. In this case, the traditional way is recording of interferograms. So I'll try to articulate on interferograms. And then the second step is the reconstruction of light itself. All right, so from this diagram, we see that the object beam of interest is combined with the reference beam. So this reference beam facilitates the integration of this property of light, which is the phase, into this captured information, which is the hologram. All right, so for the reconstruction, I would like to emphasize that the reconstruction is light itself. So meaning it has amplitude and phase information. And just to emphasize this physical quantity, which is the object beam, it has amplitude and represented by this expression AE to the I phi, and A is the amplitude. It is related, this very intuitive, no? So how much energy or how much intensity of light, from that quantity we can extract the amplitude. So amplitude is the square root of the measured intensity. Now the important aspect which is relevant to reconstruction is the phase. So as we know, the amplitude and the intensity can be captured directly, by a camera. So if we want to take a picture of our subject, we just gather the light from that subject, and that is basically the amplitude. But the phase, that is not straightforward. So the phase, I will mention several times in this presentation, represents the property of light that depends on the path traveled. In other words, it gives the shape of the light. So intensity is more of the energy of light. Phase is more of the curvature or the shape of light. So I would like to just emphasize here at this point that phase has two aspects, the temporal phase and the spatial phase. So the particular type of phase is the spatial phase, which will be the technique that we have developed is the recovery of this spatial phase information. To give a visualization of what reconstruction looks like, on the left is a video. So just to introduce how we made this video. So we took a hologram of a three-dimensional scene. So it has two action figures. And then from a single hologram, we tried to recover light itself. And this is an example. On the left is the amplitude of light. So the sequence, the frames of the video show the different planes. So at one instant, you see one object is highly focused, the other one is blurred, and then vice versa. So when the display region is where the second object is, then it will be brought to focus. Now this three-dimensional reconstruction gives us an idea of the amount of information that can be stored in a hologram, and practically how much information can be reconstructed. So it's three-dimensional. Now on the right side is the phase. So this gives us a direct access to the spatial phase information of a three-dimensional scene. So we can see that there are contours similar to like contour map in map reading in Google Maps. And each point along this fringe contour represents the same elevation with respect to a reference height. So this phase information can be obtained from a reconstructed wave. And just to emphasize, this cannot be accessed directly using an ordinary photograph. All right. So just to differentiate phase retrieval from holography. So again, in holography, you need a reference beam, and the reference beam transforms this phase information into a detectable intensity, which is called the hologram. So the drawback of introducing reference beam is that it makes the setups very sensitive to vibration. So if there is a microscopic vibration in the setup, then it is enough to wash out the hologram, and also the introduction of these optical elements to generate the reference beam makes the setup complicated. So the alternative to hologram reconstruction is through phase retrieval. So phase retrieval is the numerical reconstruction of object beam using only object intensity measurements. So in other words, you don't need a reference beam. You just need to gather the light from the object. But take note, intensity measurements. So it's plural. So it's not a single recording, but multiple recordings. But nonetheless, without the need to introduce a reference beam. So one category is called the iterative methods. And the key feature of this approach is the utilization of diversity in the object intensity patterns. So you don't just need to record multiple measurements or patterns, but the patterns must exhibit diversity. So I'll try to flesh out, now give form to what diversity means when you take pictures or measurements of the light from the object. All right, so the overall workflow will now be simpler because there's no reference beam. So you just illuminate the object, and then the scattered light, you call it the object beam. And then you take measurements or patterns, and then you feed it to an algorithm iteratively. It will extract the phase that is not directly measurable, but it is impressed upon the measurements. So therefore, there must be a way to obtain that information through this algorithm. And finally, the output is object beam. I cannot emphasize it more, but what is being reconstructed or retrieved is object beam. It is light itself. It's not just energy, but it has phase information. Now, there are different conventional strategies. You vary the wavelength, you take measurements accordingly, you vary the illumination angle, you take the object measurements accordingly, or the focus if there is a lens in the setup. So the key concept in all these phase retrieval techniques is succinctly captured by the seminal paper by Jurgenberg and Saxton in 1972, which is the first paper on iterative phase retrieval. So according to Jurgenberg and Saxton, in iterative phase retrieval algorithms, a change in amplitude distribution alone at an input plane will result in changing both the amplitude and phase distributions at the output plane. So my take on this is still very relevant, and we trying to reflect on the technique that we're developing. Here, this word alone. So it's the amplitude distribution alone. So there's no other features that you need to consider when you're recording the measurement, but the amplitude distribution. And there's another word, the change in the amplitude. So it's not just the measurements, but how the measurements change. So the relationship of the measurements is equally important. There must be a change between the measurements. All right, so the technique that we've been working on since 2006, and I still have some most of my students working on this, trying to innovate this technique. So from this schematic, we can see that the light from a coherent light from a laser illuminates your object, and then the scattered light forms this diffuse light called the volume speckled field. So it means that the object is optically rough, and then the scattered light is captured by a camera at different axial locations. And then for the reconstruction, just as an overview of this numerical method, so it's done by simulating free space propagation. So in the propagation of light, you have amplitude and phase. So the amplitude is obtained from the intensity, and then the phase is estimated, and the estimate is improved as the iteration proceeds. So just to give the basic equation, so shown on this equation is the field U at the plane P sub 1, and that can be obtained by taking the inverse Fourier transform of the product of the Fourier of the field at the input plane. So in this case, the field at P plane. So the amplitude again is from the square root of the intensity, and the phase is from the previous calculation. And then G is represents the setup, which is called the transfer function. So it depends on the distance, it depends on the pixel size, etc. And from this calculation of the field at P sub 1 plane, you can get the updated or the better estimate of the phase phi at P sub 1 plane. So the important feature here, and because I will go back to this aspect, the actual measurements must exhibit intensity variation. So you can imagine if the intensity measurement I1, I2, and so on look very similarly, then that will not be advantageous to the reconstruction process. There must be some difference, but I'll explain that later. All right, so the challenge is for the case when the test object is an optically smooth object. All right, so optically smooth means the indentation on the surface or the roughness of the surface is less than the wavelength of light. Okay, so for example, if you have a biological sample, okay, that is thinly sliced, you can imagine that upon illumination, the outgoing diffracted light, okay, will be very slightly changed only. Okay, so that is an example of a smooth test object. And of course, yung mga optical elements like lenses and other like mirrors, so they are optically smooth. So in these types of object, the generated light does not vary significantly. So therefore, this is the focus of our technique, how to investigate smooth objects. All right, so the general approach is just increase the number of planes. So instead of the typically 15 to 20 measurements, why not record hundreds or thousands of measurements? Okay, so but of course there's a limit to the physical configuration in terms of number of planes that you can measure, because you need also to move the camera. All right, so the strategy that I will present is experimental and algorithmic. So in the experimental approach, we randomize light. Okay, so instead of illuminating our test object with plane wave, uniformly illuminate with a plane wave, then instead we use a randomized light. And then the algorithmic approach is the randomization of the sequence of propagation. So as I think I did not mention it, but you proceed from first plane to second plane, second to third, and so on in a sequential manner. So the approach that we have reported is the randomized approach. So for example, you start with from the middle, and then you go to the last, and then you go to the any other plane. So there is a random, unordered sequence of propagation. I will show with some experimental results. All right, so this is a depiction of the advantage of using diffuse light. So on the left is a picture of a photoresist. So it is uniformly transmits light with very the diffraction is not very significant. And shown on the right is the plane wave illumination schematic. And then this is the result. So you can see that the three bars at the middle seems recognizable but not very distinct. So this is due to the slow intensity variation, and we consider this a poor reconstruction. So our approach of introducing diffuse light is depicted on the lower left. So instead of plane wave illuminating our test object, we first let it pass through a physical diffuser on a phase type diffuser. And then this is a profile. It is a profile scan of the diffuser. Take note that the height, the maximum height of the diffuser of the photoresist is 0.8 micron. No? And then of course it varies within 0 to 0.8. So you have this window of roughness. And then this is the sample reconstruction and well-defined edges of the bars. And this is attributed to the rapid intensity variation afforded by the diffuser. All right, so as a review of how diffuse light benefits the phase retrieval technique, we review the generation of randomized light or speckle. So on the left, we see light arriving at an optically smooth surface like a mirror. So we expect that the outgoing light preserves the shape of the incoming light. So in terms of intensity, it's the same. In terms of the shape, it's the same. On the other hand, for a rough object, the incoming light will be scattered by each point scatterer on the surface, and the superposition will produce this diffuse or randomized light. So the technical term is speckle. All right, so the parameters that will be relevant are the height, the h, and then the lateral scale sigma along the transverse direction, and then the illuminated spot, capital D. So it has been reported by Fujii and Asakura in 1974. They differentiated two types, a fully developed randomized light and a partially developed randomized light. So this will be important because the diffuse light needs to have this particular category, which is the partially developed light. So basically, the outgoing light still has information about the incoming light and the diffuse light at the same time. So it has the features of both specular and fully randomized light. So we call it a partially developed diffuse light. All right, so again, initially we were successful with the fabrication of this photoresist diffuser, but the limitation is it's wavelength specific. So in other words, it works for red light, but will fail in the green light. So it needs to have this matching of the wavelength and the diffuser property. So the challenge is to implement it with an electronic device. So we did that using a spatial light modulator. So depicted here, an incoming light arrives at the SLM, and then the outgoing light will have the special property of partially developed light. So the advantage is the convenience. Now, instead of having this wavelength specific diffuser, which severely limited, but the SLM can be controlled. So it can be adapted for a particular type of wavelength. All right, so shown here is sample measurements. Shown on the left is a specular reflection. So you can see the diffraction pattern through the aperture edge, circular aperture edge. At the middle, you see a fully developed speckle, totally random. So you can see the Gaussian distribution, but the intensity distribution is random. And then on the right, we can see the partially developed light. So we can recognize the circular diffraction, the outline of the aperture, and the diffuse light at the same time. So the reconstruction is shown on the second row, and evidently the use of partially developed diffuse light produced the optimum intensity qualities of the intensity measurements to produce the reconstruction. The test object here is the light from a lens. So we expect a spherical phase distribution, and that is successfully recovered using our technique. All right, so a few more slides. So this is the algorithmic approach. So one problem in phase retrieval is that despite the roughness of the object, for example, the amplitude is a baboon and the phase is a spherical phase distribution, there are still some local phase errors and stagnation, meaning the iterative process converges already, but if you look at the phase, there are still some errors. So this is happens in iterative phase retrieval due to many factors, object-dependent factors. So we introduced this randomized unordered propagation approach. So instead of proceeding from first plane to second, the adjacent planes, we proceed randomly. So shown on this video, let's focus on the first row and the third bottom part, top part and bottom part. So the top part, we can see the sequential manner of propagation. The middle is just a combination. So the lowest part, you see the unordered propagation. It jumps to any of the possible planes. And this is a sample result. From this graph, you can see the red rapidly approach the converge fastest. And shown on this video, on the left is the ordered or the conventional way of reconstructing, and then the one on the right, we can see the unordered sequence of propagation. So we can almost see that it reconstructs much faster than the conventional algorithm. All right, so the direction for future work, we want to develop an algorithm for non-stagnating phase retrieval using a scheduled random modulation. So here we plan to instead of doing the sequential, the different approaches like the unordered propagation, we want to perturb the measurements with some random modulation in amplitude or phase, and it has a certain schedule. So it's not just you randomize it and see if it works, but here we have this schedule. So at times it has a high modulation, and then it tapers off. So you have that. So we think that this is will provide an alternative solution to the stagnation problem. And then we also plan to use a micromirror device for amplitude instead of phase modulation with an SLM, which is significantly more expensive. The micromirror device will introduce similar effect, but can be implemented at a lower cost. And phase retrieval in a non-optical regime. So far we have done this method, implemented it in the visible, red, green. So we plan to use it in the infrared and also in the UV range, and hopefully towards X-ray with a suitable detector. So maraming salamat po. Thanks.
Thank you.
Thanks, Dux. Thanks.
Thank you very much, Bal. Thank you very much for that very interesting talk. Questions from everyone, from the audience? We have time for 2 or 3 questions.
Hi, Vince.
I have a question. Can I ask my question?
Yes, please go ahead.
Yeah, so I saw that when you implemented the Gertzberg-Sexton, you used the Fourier transforms. And the distances of your planes that you're measuring is not within considered far field, or what we call a Fraunhofer diffraction, which practically reduces the Fresnel diffraction integral into a Fourier transform. Yeah. How do you account for the errors by not performing at the far field?
Yes, thank you, Dr. Daria. So I inserted the Gertzberg-Sexton algorithm as a historical background. So, but in
your reconstruction, you still use Fourier transforms.
No, exclusively it's the Fresnel transform.
Okay, you're using Fresnel diffraction. Okay.
So it's in the millimetric range. So it's really close to the measurement planes are close to each other.
So that will take a while to reconstruct.
Well, yeah, it depends on the quality of the measurements, But in terms of efficiency, usually with around 15 to 20 measurements, we can reconstruct after 3 to 5 iterations. So 5 times 20, it's like 100 propagations, and we can see the reconstructed phase around that a number of propagations.
Thank you.
Okay. Thank you very much. Thank you very much, Bob. I have a question from Alex. Nahihiyang magsalita. What's the typical distance po between the planes, and how many planes are used usually, I guess, for the slices?
Right. So the from experiment, we haven't developed a theoretical basis for this, but from experiment we were successful. We have been successful using 15 to 20 measurements, and the distance is 1 millimeter to 3 millimeters. For the millimetric distance, it's mainly a practical solution because the micrometer stage is only 1 inch. So if you divide it with 20, so that's already almost 1 inch already. So because of that, we adapted this, and it proved to be successful. And so we since 2006 we have stuck with the 15 to 20 measurements. But now with these new numerical strategies, we were able to reduce it to around 5 to 10 measurements only. So there's a trade-off. If you reduce the number of measurements, then it will take longer time to recover the phase. If you're lucky, meaning the quality of the reconstruction, if you just increase the number of iterations, then it will reconstruct. But that's not the case all the time. So the experimental approach is to make more measurements, but it will have, of course, experimental cost. So there's a trade-off between the number of measurements and the number of iterations.
Okay, got it. Thanks, Bud. I have a question, if you don't mind, a quick one, an ignorant question, if you may say so. I noticed that you were using a helium neon laser, and does bandwidth, aside from the wavelength, actually come into play when I have a better line width, as exemplified by a helium neon laser, a very, very small wavelength spread? Would that also be quite a factor in your image retrieval quality?
Right. That's a good question, because in fact we have a paper in SPP now, using LED. There is a drawback to using helium neon laser in terms of of course, the availability and the stabilization requirement. So the alternative is to use partially coherent light. But we started with laser because of that same property of coherence. So in order to produce speckle, there's a requirement to have a coherent illumination. So the default is to use a laser, which is available now, and ubiquitous na yung laser. So it's easy to implement this technique with available lasers. But we're trying to use laser diode or LED, partially coherent light with a bandwidth of around 20 to 50 nanometer. And we have some promising results in that direction, that we were still able to reconstruct the wavefront with increased number of iterations, of course. But nonetheless, there's this promising alternative of, instead of using laser, we can use LED.
All right, thank you very much. Exciting. Okay, if there are no more questions, let me award Val with the certificate of recognition. So, Alex, ready na ba? Okay, let me read what's written on the certificate. The Philippine American Academy of Science and Engineering presents this certificate of recognition to Dr. Percival F. Almoro for delivering a lecture during the PAASE Research Expertise Cluster 7 Electronics, Electrical Engineering, and Instrumentation webinar, held via Zoom on the 13th of August 2021, signed Gisela Concepcion, PAASE President and APAM's 2020 Chair. Val, thank you very much.
Thank you,
Maraming salamat sa
Dr.
pagpapaunlak
Eustacio
sa
at
amin.
Dr. Gisela Concepcion. Thank you.
We'll send you the hard copy as soon as we get out of lockdown.
Thank you and congrats, Val. I listened intently.
Gisela.
I'm thinking of the applications. My question regarding biological samples. I'll just type it in the chat box.
All right.
Thank you, Val.
Thank you, ma'am.
Hello, good evening po, ma'am, sa inyo. All right, let's move on to our next speaker. Ready na, Alex? Okay, our next speaker is a batchmate of mine. Dr. Suzette Atienza Pabit received her BS Physics degree, magna cum laude, from the National Institute of Physics, University of the Philippines, Diliman, working on high-temperature superconductivity under the supervision of Dr. Roland Sarmago. Her interests in biophysics started as a graduate student in the University of Florida in Gainesville, Florida, USA, where she received her PhD in physics, working under Professor Steve Hagen on the protein folding problem. She then went on for her postdoc stint in biophotonics in Emory University at Atlanta, Georgia, and in small angle X-ray scattering in Cornell University in Ithaca, New York. She stayed on in Cornell as part of the experimental molecular biophysics lab of Professor Louis Pollack in the School of Applied and Engineering Physics. We call her Sushi, fondly call her Sushi, because of how cute she is. She's so cute. Hello, madam. Please welcome Dr. Suzette A. Pabit. Anytime you're ready, Suzette.
Can you hear me?
Yes. Yes.
Okay, I will share my screen. Let me. So I would like to thank everyone for coming to my presentation, and especially my colleagues, right? My batchmate from high school and college, Dr. Elmer Stasio and Dr. Val Almoro. Incidentally, Val and I also have a relationship because I knew his wife from elementary school, right? So it's a long story. But I really, really appreciate being here. I also saw Dr. Vince Daria in the audience. He was actually the one responsible for roping me in and making me join PAASE. So it is an honor to be part of the Philippine American Academy of Science and Engineering. I think one saving grace of the pandemic is that the world is a little bit smaller for us and we get to interact with each other even through things like Zoom. So today I would like to tell you about my work in the field of biophysics. I realize this is the rec is for electronics, electrical engineering and instrumentation. And even though I am in the applied and engineering physics department, my work is really more biological than what some of you might be used to. And I hope that's okay. Let me make sure I have a pointer. Does this work? Okay, so today I will tell you about our work on visualizing RNA structures. Visualizing bending of RNA structures by a protein using small angle X-ray scattering. And I will start, I just want this to be as informal as possible. So if you can't hear me or something's going on, please let me know. And I will start the talk by showing my acknowledgement slides because this is how biologists do it. This work will not be possible without a team of people helping me. And I'm very grateful to my PI, right? I'm in the lab of Lois Pollack. She always puts together an amazing team of researchers. And this work is also in collaboration with the lab of Professor Scott Showalter from Penn State. And the students and postdocs involved in the work, Dr. Yenlin Chen, Dr. PhD student Emery Usher, and Dr. Eric Cook. If you're a physicist doing biology, you need a chemist or a biochemist helping you. It's just, you can't do this by yourself. I would also like to acknowledge the Cornell High Energy Synchrotron source where we do most of our measurements. I think we are very, very fortunate to have a synchrotron on campus. And I really appreciated it when I was, stuck in the pandemic, and I realized that I'm one of the few people who can still go to work and use the synchrotron. So it was a big advantage. And our funding agencies, so we wouldn't be able to do the work without the help from NIH and NSF. So today, this is a brief outline of my talk. I will define our biophysics problem. We want to understand protein-RNA interactions, and our main goal is to actually visualize the bending of RNA by a protein. And as part of the instrumentation part of the talk, I will tell you our visualization techniques, essentially how we use physics to study biology. I will introduce small angle X-ray scattering, tell you about the information derived from SACs, and our approach that we are promoting in our lab, which is contrast variation SACs. I will maybe touch on a little bit about validation of our measurements using an optical method called FRET. And in the end, I will tell you that yes, we were successful. We were able to visualize that our protein of interest bends our RNA of interest. This work was actually published late last year. So if people are interested in preprints, I'll be happy to share. It was published in the Biophysical Journal. So to continue, I would like to define our biophysics problem. As biophysicists, we are interested in how the molecules of life work. DNA, RNA, and proteins. And this is kind of like my physicist's view of the cell. We think of the DNA as inside the nucleus holding the genetic code, and then the RNA copies the code, and then the RNA is brought out of the nucleus, and then the code is used to make protein, which are the workhorses of the cell. Our lab is very, very interested in RNA molecules, and we're finding out that RNA by itself has a lot of very important functional roles in life. Now, everybody has a coronavirus on our minds, and we know now that coronavirus has an RNA genome, not a DNA genome. And so there's a lot of things that RNA can do that we're only beginning to understand. We're also very, very much thankful for the mRNA vaccines that are helping us fight coronavirus. So RNA is very important. We're interested in how RNA molecules act with partners like proteins, DNAs, ions, everything in the cell. And in this particular talk, I focus on a particular type of RNA. This is what we call microRNA, which are functional RNA molecules. They don't get translated into proteins, but they facilitate post-transcriptional regulation of gene expression. So again, this is my physicist view of biology. In reality, biology is so much more complicated than my physicist's view. There's a lot of things going on. But I'm not a biologist, so I'm not here to describe this whole pathway. What I just want to convey here is that this primary microRNA is part of a rich area of research, which we call gene silencing. And there's big potential in terms of understanding basic biophysics here. and the possibility for novel therapeutics. In fact, this part of gene silencing is already pretty well understood. The discovery of RNA interference got awarded the Nobel Prize in Medicine in 2006. And in 2020, another gene editing mechanism using CRISPR-Cas9 got the Nobel Prize in chemistry. So, but there's still a lot of open questions in the area of gene silencing. And what I will focus on for today is this primary microRNA, or at least the very basic question of how the primary microRNA is processed by the protein DROSA and its partner to form functional microRNAs. And then it goes through its pathway, and then you get the translational repression, RNA degradation, and cleavage. Again, I won't go all through this. We'll focus on this very, very small fragment of what's going on. So this is the microRNA and it's the microprocessor complex. So in biology, people like to show this like very static pathways, but in reality, the molecules inside the cell are very dynamic. And we now are beginning to understand that conformational dynamics of RNA are very important in its function. So we are focusing on this primary microRNA as it is being processed by two proteins, DROSA and DGCR8. And the way I think about it is that, you know, the DROSA and DGCR8 work as a team to capture and then cut this primary microRNA. The way I try to explain this to my friends is that like when my son was tiny, he was really reluctant to get a haircut, like when he was a toddler. So I kind of have to hold him down, and then the hairdresser has to come in and cut his hair out. And this is, I think, what's going on with DROSA and DGCR8. So DGCR8 holds the RNA down so that DROSA can do its job to crop it, and then it can go through its pathway. So we think that the DGCR8 protein recognizes and possibly bends the primary microRNA, and then DROSA cuts it. And here we will focus on how DGCR8 bends the RNA. So in this work, we want to provide visual evidence of bending, and we do it using small angle X-ray scattering. So in SACS, we get the size and shape information of freely diffusing molecules in solution. And as a method in physics, SACS has been around for a very long time. So Val was sharing papers from the 1970s. There's this classic book by Guigné and Fournier talking about the SACS from 1955. But it's still an ever-growing field, and with the advances of X-ray physics, high-energy synchrotron sources, detectors, our ability to make better and better, more highly pure biological samples, we get to answer questions using small-angle X-ray scattering. So we do these experiments in the synchrotron because of the high flux, and we keep the sample in solution. We oscillate it to avoid radiation damage. You shine incident X-rays, and the scattered X-rays are collected in the 2D area detector. We can then do like integration to convert this to intensity as a function of Q, which is the wave vector, which is proportional to the scattering angle. And this gives us shape information. So this is actually data, our data of the primary microRNA. And this is the scattering profile, what it looks like, I versus Q. And as you can see, I have, we do a Fourier transform like Val has to do, right? Well, Val was doing a different type of Fourier transform. But we need to convert the scattering intensity into a pair distance distribution function. So we can actually look at it gives a shape information on the molecule. And I have stared at this P of R functions many different times, and you can actually tell a lot of things by just looking at pair distance distribution function. The characteristic peak here at 20 angstroms actually tells you about the kind of the diameter of the molecule, and the maximum dimension tells you about its length. And what we can do is then we can do dummy atom or ab initio shape reconstructions and try to convert this like one-dimensional profile into a three-dimensional model. So we have to try several models and then average it into what we think the molecule looks like. And then this is the average shape reconstruction for the primary microRNA. And it kind of matches our molecular dynamic simulations of what we think the microRNA would look like. It's straight, it's kind of like a cylinder, so we can kind of model the microRNA in itself, by itself, as a rigid cylinder. So the question is, what happens to this cylinder, this rigid cylinder, when a protein binds it? So now we can make a hypothesis. Again, I take you back to the pathway wherein DROSHA and DGCR8 grabs the primary microRNA. And what we think is going on is that DGCR8 grabs the primary microRNA and bends it and holds it down, and then DROSHA cuts it. So this is our hypothesis. The thing is, the question is, can we use SACs to actually see a bent RNA as opposed to a straight RNA? Before we can see if a protein bends the RNA, we have to make sure that the protein first binds the RNA. And so this is our data of like RNA alone, the black curve, and the scattering from the protein alone, which is the red curve. What you can find here is that inherent scattering from RNAs are larger than proteins because it has more electron density. So RNA has a phosphate backbone, while proteins are just made up of carbon, hydrogen, oxygen, and nitrogens. Our scattering profiles would depend on this excess electron density and the form factor, which gives us shape information. For a two-component system, if they are not interacting, you just kind of have these additive profiles. So if the RNA and the protein are not bound to each other, you expect the amplitude to be kind of somewhere here, like 0.035. But if the protein and the RNA binds together, it actually has another interaction term, which makes it light up. So you actually see a large scattering signal. So because we do everything with absolute calibration, this is a proof that we have binding or complex formation. The protein binds the RNA, and its binding is 1-to-1. So this is very interesting. We have protein bound to RNA in a regularly buffered solvent, but it's very, very hard to deconvolute what's going on the complex because the equation is kind of complicated. So one thing you can do is solve the equation, but we don't really want to solve the equation. We want to see what the RNA is doing. And so we apply a trick, which we call contrast variation. And what we do is we actually just add a contrast agent. So here, everything is in regular aqueous solvent. If you change your solvent, if you change the electron density of the solvent by adding sucrose, you can make the background to be the same as the electron density of the protein. So essentially, you can cancel out the scattering from the protein by making it invisible. So these two terms goes to zero. You have a much more manageable system. You can only see the RNA. And this is indeed what we do. So this is data of a blank protein with scattering zero. And you see already that the shape of the scattering profile is different when the RNA is only by itself as opposed to the RNA in complex. So we can do the same tricks that we did before, take the pair distance distribution function, and then we see that there is a big change in shape when the primary microRNA is by itself as opposed to with the protein core. We can do our dummy atom reconstructions again, and then there is this kink in the dummy atom reconstruction, and then we can adjust our model by using some kind of like contact approach to just like bend the RNA and say, oh, this is probably the curvature of the bent RNA after it's bound to the protein. So after going through it, I hope I was able to tell you that, yes, I think we are correct in making the hypothesis that the DGCR8 bends the primary microRNA, and we are able to visualize it using small angle X-ray scattering. We also tried to convince people further that this is really what we're seeing by looking, by validating what we're seeing using model systems. So this is pair distance distribution data on just a straight RNA double helix and a bent RNA with the helix junction helix and the shape reconstructions for those two. Another way to validate that DGCR8 indeed bends the RNA is by using a completely different approach. This is a single molecule technique called fluorescence resonance energy transfer. I mean, this is a whole talk in itself. I will not get into it, but we use it a lot in our lab to validate SAX data. Here, RNA is labeled with 2 fluorophores. And in the presence of protein, the fluorophores appear closer together. We get an increase in FRET efficiency. So I hope I was able to convince you that we were able to visualize bending of RNA by a protein using small angle X-ray scattering. And this is kind of like a demonstration of how we use physics tools to solve biological problems. And what's interesting for us is that this bending of primary microRNA by DGCR8 could facilitate cleavage by the DROSA protein. Again, we go back to our analogy of like DROSA and DGCR8 working together, these 2 proteins, like one holding down the RNA and then the other one cutting it. And I think this really has important implications in recognition and processing efficiency of primary microRNA. We know that these things are not static. They are dynamic conformational processes. And we don't know the cycle yet, but primary microRNA needs to get cleaved and then it goes through the next cycle. DROSA and DGCR8 needs to grab the second one. I believe we don't have evidence yet, but the bending actually helps with the dynamics. This is not an ATP-driven process. There's no other energy sources. So this is a very exciting discovery for us. Again, the paper has been published if you want to know more, and I am happy to answer questions. How am I on time? Thank you for listening. Maraming salamat po. And I hope I was able to share my work.
Thank you. Thank you very much. That very, very interesting talk. Questions from the audience? Yes, go ahead, Dr. Dario.
Very nice talk, Suzette. Thank you very much. As you mentioned towards the end that you don't know the cycle, and in your sample you would have a non-homogeneous distribution of bent and straight RNA. do you actually know when the time when the sample is straight and when the sample is bent? And would your pair distribution plots exhibit a combined scattering plot from both types of RNA configurations?
So from the data, since we were able to do absolute calibration, we know the association efficiency, or the KD, the dissociation constant for RNA, pre-microRNA and DGCR8. We think that based on those two things, we think that we are at least when the protein and the RNA are together, we are in the regime where 80 to 90% of the molecules are bound.
Okay.
But we cannot discount the possibility that there are free, like 10% free RNA in there. If there is free protein, the contrast agent blanks it out. So the ideal thing to do would be to model it with a straight and bent. But at this point, the only thing we can say is that since 80 to 90% of our sample is probably bound to the protein, then the bending that we see, the change in the pair distance distribution function we see, we can only attribute to the protein bending the RNA. And then there are other biochemical factors that I did not talk about. The protein actually has 2 binding sites. So there's a location for where the protein can grab 2 parts of the RNA for it to hold it. So there is, and the 2 binding sites, like just based on like geometry, it can't be straight. It has to be bent for this to happen.
But from my question is basically, if you know the exact cycle, would you be able to clearly identify a sample that's purely straight and a sample that's purely bent?
I think we will always have some, like, I think our error is still like 10%. So we don't, unless we actually go to, no, I don't think we have like that level of control, which is always unfortunate when you deal with biological systems.
Yeah.
So you can kind of like make assumptions on what is going on, but it's always there's always like a possibility that there is a fraction that's not that's straight. Yeah.
Thank you, Vince. Thank you, Suzette. Any other questions? Yeah.
May I ask a question?
Yes, please, Ma'am Giselle. Just go ahead.
Sorry. Sushi. It's just wonderful work. And I'm just curious how this is a refinement of more traditional high-res X-ray crystallography, where we actually can, at high resolution, show the binding of a protein with a small molecule, maybe smaller than a microRNA. And what's the advantage of this technique of the angle where you can show the bending? That's my first question. So we know very well that in many cases, it's really better to get the big molecule, a protein, in complex with the smaller molecule. Okay. Second question is that in reality, so you think you're like close to 90% of the true picture, but am I right that you isolated the protein and the microRNA, or had them synthesized, maybe in the presence of the second protein, which I presume is an enzyme because it cleaves, it's a nuclease. Were those used in biologically relevant concentrations, because the problem always is when you study the interactions and the structure and the interactions of these biomolecules. The rule is actually, as demonstrated by you, promiscuity, multi-protein, and other biomolecule interactions. So how close is your model to the reality? Okay, and then third is, you mentioned the big K, which is the K of binding. That is just binding, but then is there a quantitative measurement of the rate of curvature or the rate of change, or the rate of change of the bending? Because that would be critical to regulation and function, and that's what we're trying to elucidate. And best known systems are, of course, enzymes and their substrates, where the substrate can actually be a small molecule, an organic molecule, or another protein. And there's always the big K and the small k, which is for the catalytic function. So I'm just curious whether you are moving in this direction. Crystallography, as I understand it, pertains to structure and binding, but then function is energetics, okay, bioenergetics. But then as biochemists, we always look at catalytics or rates. So I'm just curious in what direction you're headed. Thank you.
Oh, I love your questions. That's a lot of questions. That's a lot to unpack. It's very, very, very important. I think crystallography is still the holy grail. Because small angle X-ray scattering, because it's essentially a low resolution technique. We get 10 to 20 angstrom resolution. While if you can have a crystallized molecule, you can get to like 2 angstrom resolution and actually nail down what is going on. One problem is that these proteins, DGCR8, is very, very hard to crystallize. So not all proteins can be crystallized. And so it is harder to understand. If you can't turn it into a crystal, then it won't diffract. You can't get the crystalline resolution that you would need. With DGCR8, it has a lot of intrinsically disordered regions, which makes it really hard to access in terms of crystallography. And that's why we are trying to use a more low resolution technique like small angle X-ray scattering to look at like size and shape information. There are, so my laboratory also works on like crystals. We are not at the moment trying to crystallize DGCR8. What we're looking for are dynamics. And when you have a crystal, it's harder to probe dynamics. When you have a molecule that's freely diffusing in solution, you can do other fun things to it, like change the solvent rapidly using time-resolved mixing experiments and actually see how the conformational change happen in real time. So that is one of the, I hope it answers your first question. There is a published crystal structure of Drosha by itself, but it doesn't have the DGCR8 part, it doesn't have the RNA part. So getting all these complexes together is a feat in itself. And so we're still trying to figure out what this all means. So there is a fragment of DGCR8 that is crystallized, but like there are in little pieces. So as we put together the story, and again, this whole pathway, maybe we can understand what's going on. What we were able to do though, in terms of our modeling, since we have an envelope, we were able to reconstruct the envelope of what we think the dummy atom reconstruction of the bent RNA is like, is we were able to actually postulate what the bending angle of the bent RNA is by doing kind of like a matrix rotation of the all atom molecular dynamic simulations of our straight system. So how it plays into the bigger role of DROSHA and DGCR8 and the pathway, we don't know. So it's kind of like every little piece of research you do, you actually like open a little window of understanding, and then you open like another kind of worms of all these questions. And so we're only beginning to understand these details. And as a physicist, I have to work closely with biologists because they are the ones who are helping us make the samples, and we are the ones like trying to see how we use our techniques to be more quantitative in our problem solving. So a lot of the innovation we do in the lab is like, you know, what I told Vince, we need absolute calibration, we need pure samples. And even so, even if we know we're starting from really good samples, there's still a lot of questions. But slowly, you build a story until you have a big picture. Because no molecule is alone. Like molecule A has to bind molecule B. And we need to understand how they come together, how they fall apart, so that we can like keep understanding, keep getting better in having quantitative understanding of these processes. Did I miss any of your questions?
I absolutely appreciate your answer, Sushi, and I share your excitement in pursuing this project, which could take a lifetime. And I just wanted to say that, you know, in classical enzyme kinetics, there is this creation of torsion or tension or bending. The lock and key or induced fit is something that you seem to be seeing here. And I'm just curious, like there's the active site, and then the point of bending, or the angle, the point of bending, is that where the cleavage happens? I'm just curious.
Yeah, the cleavage happens elsewhere.
Oh,
and
okay.
what we find, there are sites for cleavage.
Yeah.
And so there's other things. It's like we
Oh.
think that the bending of DGCR8 is promiscuous. It doesn't care if it's a primary microRNA. As long as it sees a double helix, it will bend it. RNA.
Okay.
So there is something else so if you give it a primary microRNA with characteristic sequences, it will bend it, but it will take DROSHA to have the recognition mechanism to actually cut it. And we don't know what's going on in the nucleus because there's a lot of molecules, right? Does
exactly.
the DGCR8 grab just anything randomly, and then does DROSHA reject it? We don't know. And then what's the processing efficiency? What's the hit rate? There's a lot of questions. And since we are beginning to use physics to ask these questions, it's very hard for a physicist to work on these very, very messy systems.
No, you have the capability to learn.
But we have to, right? We have to, which is why it's nice to look at these problems.
Thank you so much, and congratulations, Sushi. Yeah, we'll follow your work. Okay, thank you.
Thank you very much. Thank you very much, ma'am. Thanks, Suzette. Yeah, I guess we probably should be wrapping up. Medyo papagabi na kina Suzette at this time. Thank you, Suzette, for your time. Bob?
So thank you, Dr. Pabit, for sharing your research to PAASE and the science community. So PAASE presents the Certificate of Recognition to Suzette Atienza Pabit for delivering a lecture during the PAASE Research Expertise Cluster 7, Electronics, Electrical Engineering Instrumentation webinar, held via Zoom on August 13, 2021, signed Dr. Gisela Concepcion, PAASE President and APAMS 2020 Chair. Thank you, Suzette.
Maraming salamat.
Thank you very much, Suzette.
All right.
Thanks. All right, let's move on to
the
next speaker, Bob.
Okay. So our next speaker Dr. Maria Herminia Balgos is a postdoc researcher at the Innovative Photon Manipulation Research Team in Riken Center for Advanced Photonics in Waco in Saitama, Japan. Dr. Balgos obtained her BS Applied Physics, MS Physics, and PhD Physics at the National Institute of Physics in UP Diliman. Also, she worked as a UP faculty as a graduate student. Her PhD dissertation was on spatiotemporal characterization of electrical and optical properties of low-temperature grown gallium arsenide structures for terahertz applications. Her current research are on phase detection techniques in both near-field and far-field regions of the electromagnetic spectrum. Ladies and gentlemen, Dr. Balgos, you may start.
Hello, morning everyone.
Yeah, good morning.
Good evening, Ma'am Suzette. So first I would like to thank the organizers for allowing me, giving me the opportunity to talk about our work. So my talk is about the scanning, tunneling, microscopy, and spectroscopy of gallium arsenide terahertz emitter. This work is in collaboration from 3 institutes. So we have the Surface and Interface Science Laboratory at RIKEN, the National Institute of Physics in UP Diliman, and the FIR Center at the University of Fukui, Japan. So the photos here are my collaborators and my co-authors for this particular paper. So just a brief overview of what we wanted to do. We know that the introduction of interface has been shown to affect the terahertz emission of gallium arsenide. In fact, our group has presented a lot of papers already as to how the introduction of interface is. Now, however, most of our works, or in general works on gallium arsenide, do not have the direct probe of the interface. And so we would like to challenge ourselves to directly probe the interface using a scanning tunneling microscope, or STM, and correlate it to the terahertz emission intensity. So just for a brief background, the terahertz emission in gallium arsenide is due to the ultra-fast photocarrier drift at the surface or at the interface of a gallium arsenide emitter. So for example, you have an ultra-fast laser source impinging on the surface of the sample, then electrons and hole pairs are generated. So the electrons move towards the surface and then the holes move towards the bulk. Now this ultra-fast drift current results in a terahertz emission, and then this drift current is proportional to the built-in electric field either at the surface or at the interface. I would just like to note that often high resistivity gallium arsenide is utilized as a emitter. So for the technique itself that we used, we used the scanning tunneling microscope, or the STM. So it's basically just a microscope that can resolve up to an atomic resolution. So the STM was invented by Binig and Rower in 1982, and they were awarded the Nobel Prize for that. So essentially, the scanning tunneling microscope takes advantage of the tunneling electrons, quantum tunneling of electrons within a gap. So for example, you have here your tip, metallic tip, and then you have your metallic samples. If a bias is applied across these two electrodes and the distance is small, such that quantum tunneling occurs, electrons can tunnel from the tip to the sample and then vice versa. Now, the very high resolution of the STM rests on the strong dependence of the tunneling current with the distance d. So this is given by this equation. So the tunneling current is proportional to the exponential of d. So what this equation means is that very small changes in d would result to high changes in I. And thus, if we try to scan at a constant current, then the tip can actually map the topography of the sample under test. Now, this was the very first STM image from this study. So from here, they said they can see the mono, double, and triple steps, mono steps, and they also were able to image gold surface. Of course, since this was the first one, it's not so good when you look at more previous images of STM. So STM, in using STM, we are presented with two challenges for our purposes. So the first one is that because the tip and the sample must form a closed circuit, normally the sample is limited to conducting surfaces. If the sample is non-conducting, often you will not detect any current and then the tip would just crash on the surface. And then the next one is that this technique is limited to probing few atomic layers only at the surface. So we wanted to address these two main challenges for our purposes. The first one is to probe a layer with low conductivity. So as I have said, for terahertz emission, high resistivity material is often preferred. So what we did was to deposit the low conductivity layer on highly conducting substrate to improve the overall sample conductivity. So this was the sample grown by Dr. Prieto, and this is a 2-micron high resistivity gallium arsenide. So this is the layer with the low conductivity. And then we deposited it on a high conductivity substrate buffer system. And then to probe the interfaces of the thick samples, we exposed the interface through mechanical cleaving. So what we did was to get the sample cross-section, and that is where we tried to scan the tip. So also, I'd like to note that we did the cleaving in vacuum to minimize any sample contamination. So this is just a photo of the actual sample. This is the uncleaved sample. And then what we do is put a scratch here, and that would be initialize the cleavage. So in vacuum, I used just a wobble stick to push the remaining, to push this part, and then it would just cleave itself. So after cleaving, actually, you just pray to God that it's flat. And so this is just the photo of a cleaved sample. And then, as I have said, we scan across the sample cross-section. Now, for probing the undoped gallium arsenide or the high resistivity layer, the keyword I would like to use is the diode under forward bias. Now, this is just what the sample schematic is. So you have the tip here, and then you have the sample here, and in a common ground. So when you do a positive sample bias, the free electrons in the N gas moves toward this area, and also the applied field is in the same direction as the junction electric field. So suppose you have already an initial electric field, and then applying the positive sample bias increases that built-in electric field, and so it suppresses the movement of the carriers from the N gas to the LT gas, and then vice versa. Now, at negative sample bias, it's the opposite occurs. So the free electrons move towards the LT gas. And then also, because the applied electric field is opposite the direction of the junction electric field, then the electric field at the interface becomes slower, and that permits more carrier movement across the interface. Now, so of course, since we were dealing with homojunctions, as you can see here in figure C, it is very difficult to see which one is the gallium arsenide, highly doped, and the low resistivity gallium arsenide. So the topographic contrast between the homojunction is not distinct. So the different steps here are just monoatomic steps in the surface, and is not at all indicative of whether or not we are at the LT gas layer or at the substrate buffer layer. So because the tunneling current depends on the bias voltage, we wanted to see the difference in the carrier concentration, which affects the DIDV, or the differential current voltage. Okay, so to locate the interface, I follow this approach. So first, I start at the very close to the substrate and then move my way towards the LT gas. Now, if I see any difference in IV, then I think that the interface is found. Of course, I do some tests afterwards, and then, but if not, I just continue moving towards the LT gas direction. So this is an example of the DIDV curves taken at the positions indicated by the dots here. So I color-coded them into red, green, and blue. So at the red, we have the high DIDV at positive bias voltage, as you can see here. And then the green one, we have the comparable DIDV at both the negative and the positive sample bias. And then lastly, at the blue curve, we have only high DIDV at negative sample bias. So this is actually indicative of what the doping, or the doping concentration and the doping type of a certain layer. Now, the high DIDV at positive bias signifies an N-type, or an electron-rich layer, while the opposite, the blue ones, indicate a P-type, wherein you have an excess of holes. So because of this one, using this result, we were able to identify where the interface between the LT gallium arsenide and the N-type buffer. However, because we use the same doping concentration between the substrate and the gallium arsenide buffer, we cannot identify that particular interface. So this one here is just an estimate of the location of this particular interface based on the nominal thickness from the growth, MVA growth. So next up, we look at the smaller area of the layer. So we have here an N plus gas and LT gas. So as you can see, there is a little bit distinction between the N gas and LT gas right at the interface in number 5 here. So we can see, looking at number 5, we see that there is a decreasing current, tunneling current at positive sample bias. And then a similar paper before that, taken from silicon with decreasing dopant intensity, suggests that this area here, 5, 6, 7, and 8, we still have N carriers, but then they are decreasing as we move towards the LT gallium arsenide layer. Now, using this result, we were able to map the electron density at the interface, close to the interface. So we start with the nominal 10 to the 18 doping concentration of N gas, and then as we move close to about 18 nanometers, the electron density decreases by an order of magnitude. And then at around 50, or I think 40 nanometers, the electron density is already at 10 to the 15, which is a characteristic of semi-insulating gallium arsenide. So from here, we were able to suggest that close to the interface, the free carriers from the N plus gas affects the electronic property of the LT gas. So very close to this area, very close to the interface, there is still the presence of positive tunneling current because there is a carrier transfer from N gas to LP gas. Now, far from the interface, the LP gallium arsenide becomes semi-insulating already, and then we do not observe any tunneling at positive sample bias, as can be seen from number 8 and actually also number 9. Now, looking at a more closer area, this is very close to the interface already. So this is the topographic image where I took the DIDV map here. So each each vertical line here is one atomic row of arsenic. So as we can see here, there is a transition from N to P around 15 nanometers from the interface, similar to what we modeled from the previous slide. And then we can see here that the trend of the DIDV signal at positive sample bias is characteristic of the electronic band structure. And so from here, we were able to say that we had a real space visualization of the band structure and a direct probe of the interface. And so using our results from the STM and the STS, we were able to approximate the electronic band structure of our sample like so. Okay. And so now this electronic band structure is actually the main reference when we wanted to explain the terahertz emission. Now, what we did was to vary the sample thickness. So essentially, we have a starting with 2 micron LT gallium arsenide. We did a wet chemical etch, using this particular etchant, and then we were able to vary the thickness of the layer. So we also did the same thing for a semi-insulating gallium arsenide substrate that would serve as our reference. Now, based on our measurements, the etch rate is about 8.61 plus minus 0.97 angstrom per second. And of course, because terahertz emission is also sensitive to the surface morphology, we also did some AFM measurements and Raman spectroscopy for the crystal quality to ensure that we did not destroy or alter the sample through the wet chemical etch that we did. So these are the AFM micrographs. So we can see here that all of the samples we used have comparable surface roughness. And then this is the Raman spectra where we have the allophonon mode here. And as we can see, although there is a difference between the intensity of semi-insulating gallium arsenide and our LT gallium arsenide sample, the etching that we did not affect the line width nor the intensity of the allophonon of gallium arsenide. And so from here, we can conclude that these samples have comparable quality of the surface. And so now we are only left with the interface effect. Now, this is our results for the terahertz emission. So the first one is from the Si gallium arsenide where we do not have any interface. So as we can see here, the terahertz emission does not vary with the thickness of the semi-insulating gallium arsenide. Whereas for our sample, we found an increasing terahertz emission from 2 micron and then up to 1.04 micron. We also like to note that there is no change in the bandwidth for both samples. Now, we wanted to explain the variable thickness terahertz emission based on the electronic band structure that we were able to get from the STS. Now, for semi-insulating gallium arsenide, because there is no interface, the electronic band structure remains the same regardless of the thickness. And so we do not see any difference in the terahertz emission intensity. However, for the LT gallium arsenide, we can see a drastic change. So this is for the 2 micron 1.5, and 1 micron. So as we can see, there is a change in the electronic band structure with thickness. So the built-in electric field is, we can imagine it as proportional to the slope of this area here. And as we can see from the 2 micron versus 1 micron is that the electric field at the LT gas, across the LT gas surface, is much higher compared to the 2 micron sample. And thus, we have the most intense terahertz emission for this particular thickness of LT gallium arsenide. And then lastly, we looked further at the pump fluence dependence of terahertz emission. Now, if we want to use this as an emitter, we have to consider by how much can we actually, by how much power of laser can we use before saturation of emission occurs. As you can see here, for the semi-insulating gallium arsenide, it is linear with pump fluence, while that of the LT gallium arsenide increases at lower pump fluence and then eventually saturates at a higher pump fluence. And then we would also like to note that the saturation is highest for the 1 micron sample. Now, there are two causes for terahertz emission saturation. The first one is the near field screening effect, and the second one is the column screening effect. Now, the near field screening effect occurs when the sheet conductivity exceeds the threshold. For gallium arsenide, it is 12 milliseconds. Now, semi-insulating gallium arsenide has a higher sheet conductivity and higher mobility than our low temperature grown gallium arsenide. And thus, we expect it to saturate at a lower fluence. So clearly, the near field screening is not the reason for our saturation, for our observed saturation. Now, for the column screening, it is caused by the spatial separation between the carriers, and this becomes very efficient when there is a static built-in electric field at the interface, because there would be an efficient trapping of electrons. Also, we would like to note that the high quality of the interface increases the recombination. So from the STM image, we did not see any abrupt changes in the topography. And so we think that the recombination at the interface is quite efficient as well. And because of that, the built-in electric field is reduced when the next pulse, optical pulse, arrives, and that causes a saturation of the terahertz emission. And since the 1.04 micron has the highest built-in electric field, it also has, consequently, it also has the highest lowest saturation value. Okay. So as a summary, so STS, we showed that STS is effective first in locating the interface between the homojunction. So we wanted to iterate this one. And near the interface, the LT gallium arsenide has an N-type conductivity due to the free carriers injected from the buffer area. And then away from the interface, the LT gallium arsenide becomes semi-insulating. And then the terahertz emission efficiency of the layer is correlated to the electric field within the entire gallium arsenide layer. And then lastly, the high quality of the interface and the free carriers results to a saturation of the terahertz emission at high optical pulse fluence. Now, of course, more information can be found in our paper related to in this link, shown in this link. So I'd like to acknowledge, so this work is supported in part by grants from these bodies. And then also this research was done during my PhD stint at RIKEN. So I would like also to take this opportunity to thank RIKEN's Program for Junior Scientists and the National Institute of Physics. And then the STM, preparation of the STM tips is from Yoshiko Shimizu and Yuki Hasegawa. And I would also like to thank my current boss, Dr. Tanaka, for allowing me to present for today's meeting. Thank you very much for your attention.
Thank you very much, Mimi. Questions from the audience? We have time for 2 or 3 questions.
Professor Estacio, I have a question for Dr. Balgos.
Go, Alex.
Hi, Ate Mimi, Dr. Balgos. Thank you very much for the very nice presentation. I have one question. In your scanning tunneling microscope experiments, what determines the spatial resolution?
Oh, well, the spatial resolution is determined by how good your tip is. So the more the tip, because tunneling has to be confined at the most atom, at the atom nearest the sample. So if the tip is blunt, not so sharp, then the spatial resolution becomes worse. So that one. So before we actually do these experiments, we have to do a lot of, we call it the tip making. So it's essentially just do random things on the surface of another sample and then have it, try your very best to make the tip as sharp as possible.
And the tip is made of what material?
Sorry, I forgot to mention, for this particular work, we used a tungsten tip.
Okay, thank you very much.
Yeah, Mimi, just to follow up on Alex, aside from the quality and the dimensions of the tip, would the material characteristics of the sample itself give an ultimate resolution and maybe penetration depth for the imaging? Like the more conductive the gallium arsenide would be, the better, and the better resolution you would have, the easier, because this would make the sample easier to bias and would also have a, I don't know, maybe deeper penetration depth. And if you have a semi-insulating sample, then it would be more difficult to get the desired resolution that you would otherwise expect from this technique.
Thank you very much. So actually, baligtad po. It's quite the opposite. So when we do at least maybe it's just my technical skills, but then it's very difficult. It's more difficult to get atomic resolution from conducting samples, because we are working with tunneling of electrons. So essentially, it's not the real topograph, but actually a map of the density of states. Now, if you have a metal sample, the density of states is not so flat. It's a little bit more flat compared to when you have a semiconducting or an insulating material. So it's quite the opposite. The downside only for insulating material is that you need to use a very large sample bias in order to observe tunneling. But once you observe that, based on my experience, it's quite easier to do the atomic resolution imaging for not so conducting materials.
I see. All right. I understand. Thank you for the clarification. So you can work with the semi-insulating down to how much doping concentration have you tried working on? What's the upper limit, if there's any?
Upper limit, sir, of doping concentration?
That you tried, just experimentally.
Yeah.
Have you tried 10 to the 18?
Yes, this one, we have 10 to the 18 for this particular sample. And then when I was teaching myself how to use the STM, we used 10 to the 20 also.
Okay. Which would definitely affect the crystalline quality of the sample and would have shown by STM.
Yes.
All right. Okay. Thank you very much. Very interesting and also very, very expensive.
Yes, very expensive.
Research area.
Time consuming also.
If it's okay, would it be possible to give us a ballpark figure? How much would an STM initial capital outlay, including maintenance, would be? I suppose this would have a lot of cryogenic accessories that need to be maintained. if you want to operate it on a regular basis?
Yes, so actually, sir, the STM I use for this particular study is a Omicron. It's a commercial STM from Omicron, and it's very costly. I am not so sure about the ballpark figure, but maybe hundreds of millions of yen for one system.
That
And
would still
then,
be hundreds of millions of pesos.
yes.
Okay.
And
All
then
right.
maintenance itself is also quite expensive because we use helium to lower the temperature down to 4 Kelvin. And then we have to refill the helium tank. And so it's not like a closed cycle helium cryostat. We have to refill every time.
Yeah. Thank you, Mimi. Yeah, to put things in perspective, the two speakers that we had today who came from abroad, Mimi and Suzette, work on very, very expensive toys. And yeah, in the context of the Philippine setting, it would be very, very advantageous for us here in PNAS to engage in collaborative work if there's an opportunity for us to be able to gain access to such seemingly, at this point, impossible, almost impossible way to have access to these kinds of high-end research equipment. Suzette uses a synchrotron. A synchrotron facility is itself very, very expensive to maintain. So yun, maraming salamat, Mimi. Just a moment. Let me read the certificate of recognition. The Philippine American. Yes, Doug, sorry, you have a question.
I have a question, si Dr. Daria.
Sorry, Vince. Please go ahead.
All right, so I must have missed it, but I saw that the interface between the two semiconductors, if the interface is not perfect, would you get would you be able to detect that from the STM? And how would that affect in terms of functionality in terms of generating terahertz, for example?
Yes, actually, sir, that's a very good question. Yes, if there is a defect at the surface, we can see.
Not just as a surface, at the interface between the two semiconductors.
Yes, we would be able to see that. If there is a topographic contrast, we can see it via STM. So,
and does that correlate with the function of the terahertz in terms of intensity or how much efficient optical efficiency with it?
yeah, sir, I think if the interface is not so smooth, I think there would be a lower terahertz emission because then the recombination would be very efficient at the will be localized at the interface because the interface is not so smooth. And so because of that, the current, sorry, the photocurrent drift will also be reduced and the terahertz emission will also be reduced.
But how would the tunneling image would look like? How would the current from the tunneling microscope look like in terms of profile?
So the tunneling current would, those scans that I presented a while ago is a constant current mode. So we maintain the tunneling current. And so because the difference in the, and then change the D or the distance between the tip and the sample. So in terms of the...
My question is actually, can you actually monitor that from the tunneling current, from the tunneling maps?
From
a defect on the interface.
Hi, yes, sir. So I have few samples like so we can actually see those defects.
Okay. Okay, Navin.
Yep.
All right. Okay, thanks, Mimi. So let me, if there are any more questions from the audience that I may have missed, pasensya na po. Okay. The Philippine American Academy of Science and Engineering presents this certificate of recognition to Dr. Maria Herminia M. Balgos for delivering a lecture during the PAASE Research Expertise Cluster 7 Electronics, Electrical Engineering, Instrumentation webinar held by Zoom on 13th August 2021, signed Gisela P. Concepcion, PAASE President and APAMS 2020 Chair. Thank you very much, Mimi. Salamat sa pagpapa-unlock. All right, so it's 10 o'clock. We have 5 minutes to spare. Maraming salamat po sa lahat ng dumalo. Kami po ni Val ay natutuwa. Pasensya na ito supposedly would have been a July webinar, but due to unforeseen circumstances, pasensya na po, we had to reschedule it a month after. So thank you very much for all your support. Thank you very much, PAASE. Val?
Yeah. Thank you, Dr. Concepcion. Nandito si Dr. Serapica also. So the PAASE provides a synergistic relationship among the researchers. And I think we have a critical mass of Pinoy scientists. So hopefully we can have this similar webinar in the future. Thank you, Dr. Estacio.
Thank you very much, Val. Thank you very much, Alex. Thank you, everyone. Maraming salamat po. See you next webinar. Okay.
Well, worth the wait, Elmer, Val.
Maraming salamat po.
Maraming salamat. Thank you.
Pwede ba tayong attendance picture if it's okay? Some people have left, but pasensya na nakaligtaan ko. Okay lang po ba, ma'am? Wouldn't take 2 minutes.
Of course.
Thank you po. Thank you po. If you